Hey everyone, so today we're going to be talking about DNA replication. We have the theta model kind of drawn on this whiteboard here. We're going to be looking at this a little bit more in depth as to what's going on truly around the replication forks and all the proteins that go along and help DNA replication. Hey everyone, so this is going to be our up-close DNA replication. We've got our 5' 3' ends and the same on the other side. Up top we have our leading strand, and we know that because it's continuous and it's replicating towards the replication fork here. And then our lagging strand is replicating away from our replication fork, and it's got these fragments. It's discontinuous replication. So we've got some household items that is representing the proteins that are in play here. So we've got the little lifesaver that's representing gyrase, which is a topoisomerase that relieves supercoiling ahead of the replication fork. And what that helps with is it helps then helicase come in, which is represented by the star with the H on it, to come in and help break hydrogen bonds that exist between DNA base pairs. And that helps open up this bubble of replication, and it uncoils this DNA that's coming in right here. So next we have our little pair sticker, and our pair sticker represents primase, and what DNA primase does is it synthesizes short RNA primers that allows DNA to begin replication. So these googly eyes right here, those are the little primers that exist on the end of this DNA lagging strand. And these fragments are called Okazaki fragments. They're just fragments of DNA that on the lagging strand. And then what we've got here, this yellow pom-pom, is DNA polymerase 1, which is actually going to go in and remove those primers that primase put down, so that later ligase can come in, which is here, which will come in here, and it'll fill in these things called nicks, which are like the little gaps that are created by the Okazaki fragments. And so what we've got with our little tiny stickers here is single-stranded binding protein. So what that does is it coats single-stranded DNA and helps stabilize it and protect it. And it also prevents it from forming hairpins, which is really important. We don't want any hairpins in replication. So finally, we've got our little red pom-poms there and there, and that's going to represent DNA polymerase 3. And that is essentially what synthesizes our DNA. It's adding nucleotides here and here where replication is moving towards, from the 5' to the 3' end. So yeah, we've got, the last thing I want to talk about is phosphodiester bonds. Those phosphodiester bonds are what exist between nucleotides, and they help connect the sugar and phosphate groups. Ligase here actually will come in and add a phosphodiester bond. It doesn't add a nucleotide, it adds a phosphodiester bond in where the little nicks occur.